Hello, good afternoon, everybody, and thank you, Dr. Renaud and ULF for the opportunity to give you an update on Vigil and our program in ALSP. For those of you who don't know me, I'm Ivana Magotrić-Libiš, the President and CEO of Vigil, and it's a real privilege to have spent the last few days with this incredible community. So let me start by telling you who we are. So Vigil is a clinical stage microglia-focused therapeutics company. Our goal is to discover and develop novel therapeutics to treat both rare and common neurodegenerative diseases by restoring the vigilance of microglia. And you've heard a lot about microglia in the past few days. They're the brain immune cells. They're responsible for maintaining the health and well-being in the brain, and they control a series of neurological functions to prevent neurodegeneration. As Dr. Eichler said earlier today, they're the ones that go out there and remove the damage in the brain. So really important cells. And what we have identified is that TREM2 is one of the main control switches for microglia function. It is a receptor that sits on the surface of microglia, and it's the main damage sensor in the brain. So we actually are the only company that actually has two different TREM2 molecules. We have both a fully human monoclonal antibody TREM2 agonist as well as a first-in-class small molecule. Today we're going to be focusing on our antibody because that's the molecule that's in clinical trials for ALSP. The other thing I think that differentiates us a bit is that we really take a precision approach to neurodegeneration. What that means is that we're really focusing on diseases where there is a very direct genetic linkage to microglia dysfunction, because we believe that this will allow us to actually get to data quickly and ultimately reach the patients as quickly as possible. We have been around only for about 4 years. We actually have our 4th-year anniversary coming up on July 10th, but the team has accomplished a tremendous amount, and as I said, today though, we're gonna just focus on ALSP. So because we have these two TREM2 modalities, even though it's been such a short time frame, we have built a really robust pipeline at this point. As I mentioned, the most advanced program is the program in ALSP. We actually have the antibody, which we call iluzanebart, or I'll call it IBART for short, which is currently the first interventional study for ALSP. We also have a natural history ongoing in ALSP, and I'm sure, again, you've heard a lot about natural histories in this past few days and the importance of them in really trying to understand the disease and disease progression. So I'll be spending a little bit more time on that as well. And then our small molecule, VG-3927, is actually also in the clinic, but right now in healthy volunteer studies, and we believe that it has a potential to be the next therapeutic for treatment of Alzheimer's disease. So now let's switch to, I don't know what's happening with the slide. Okay. Yeah, the slide is, all right, we'll just skip. So basically, let me focus on IBART. So what I want to talk to you about is our natural history study, our phase 2 study, share some of the data as well, and talk about the next steps. So as I mentioned, critical for diseases where we're trying to understand what is happening is to conduct a natural history study. And so this study has actually been going on now for over 2.5 years. It has enrolled approximately 50 patients globally. And what we're really trying to understand here is, what does the disease progression look like? What are the changes that we're observing? And can we identify biomarkers and clinical endpoints which we can then use in the trial design to really understand whether our molecule has efficacy in this condition or not? So this is a critical study. It's a critical part of our clinical development plan, and what we're looking at here is MRI as one of the main biomarkers of the disease progression. We also recognize that in this patient population, NFL levels are highly elevated as well, so we're looking at that. And we're also, what's really exciting is that we're actually starting to see a correlation between the changes we observe in the MRI in the patient population and the disease progression. And obviously this is going to be critically important when we start our conversations with regulators, because we know that this disease is so fast progressing that we don't want to have a study where we're waiting for clinical outcomes. What we want to do is be able to use biomarkers as surrogate endpoints and really try to go for accelerated approval, because the idea is to get to patients as quickly as possible. So very critical study for us, and as I said, we're getting some really good data out of the natural history study, and we also want to use the natural history study as a potential synthetic control arm. As you guys heard a lot about placebo-controlled trials, we think that in ALSP patients, it's going to be very challenging to conduct a placebo-controlled study, and so we want to use the natural history as a synthetic control arm to avoid that as well. So in terms of the study, as I mentioned, first interventional study in this patient population. It's the study that's currently ongoing. It's really excited to say it's fully enrolled, and we're very, very grateful to the LSP patient community for stepping up and enrolling in the study. The study is 12 months in duration. It's a monthly IV infusion of the IBART, which is, as I said, is a fully human monoclonal antibody. We have 2 doses, a 20 milligram dose and a 40 milligram dose. And what we're looking in the study, obviously, primarily is safety and tolerability, but also looking at how these biomarkers move. And at 12 months, we'll also be looking at some of the clinical endpoints that we have agreed with the agency on. So we did have a first interim look in this study, which was in last November. We actually took a look at 6 patients at 6 months. Obviously, that's very early in the study, and the numbers are small, but we're seeing some very encouraging data even this early on. We're actually seeing slowing on progression on some of these biomarkers, mainly MRI and NFL. Most importantly, the drug is very well tolerated. It's safe. We have not had any A's or SA's, which is great to see. And also very importantly is, as I said, this ability to take a look at biomarkers and then put them in the context of what we're learning in the natural history study. So there's more to come. The next readout from this study is going to be in the 3rd quarter, so not too far away. And then what we'll be looking at is these patients 12 months out, which is actually the actual duration of the study. And then we'll be looking at the 40 milligram dose, which is the next dose, at 6 months. And then ultimately the study will end when all patients have 12 months of data. However, we're allowing patients to continue on drug past that 12 months. So we do have patients who have finished the 12-month and are on the what we call an extension. So really, really encouraged by what we've seen so far, but obviously a lot more to come. So I've mentioned biomarkers several times, and I know a lot of people talk about biomarkers and why biomarkers are so important. And as I said, the reason for that is that we want to start the conversations with the agency about what's called accelerated approval. So the ability to actually get the drug approved on biomarker changes. And so right now, we feel that with the natural history study data and the data we already have from the phase 2 study, that we have enough information to engage the agency. We're actually very, very grateful to the Sisters Hope Foundation because they had a very successful patient-led FDA listening session in January, where they started to educate the agency about the disease, the severity of disease, the fact that it's so devastating and so fast progressing. So that's extremely helpful because the agency's never heard about this disease before. So all these advocacy efforts are very helpful when we go and start these conversations with the agency around the biomarkers. Now, obviously, regulator is very important, but what's really important is the engagement with the patient community. And we really do believe that that engagement needs to happen very early in the process, and the voices need to be heard. So from day one, we have engaged the patient community, the KOLs, the families, patients, caregivers, all of you, to make sure that we really understand the disease, really understand the patient journey, really understand where are the gaps in knowledge and understanding, so that when we go and conduct these studies, we are armed with the right information to make sure that we are looking at things that actually are going to help us determine whether there is efficacy or not. So we have been talking to the community, and one of the things that came across loud and clear is the need for cost-free genetic testing. So we know that this disease is misdiagnosed and underdiagnosed. So it is not something that when you go and see a doctor, it's going to immediately come into mind. This disease looks like a lot of other diseases such as MS, FTD. So it was really critical to make sure that people are thinking about ALSP, and when they are not sure about the diagnosis, they have an opportunity to test. So in addition, this program is called ALSP Aware, and in addition to providing it to families, so free testing and free genetic counseling, we're also actually providing a test to the healthcare community, really focusing on areas of misdiagnosis, and for example, in MS centers, so that we can make sure that the patients are identified as quickly as possible, because we all know, we've all talked the past few days about the need to go and treat early, and that's same in this disease as well as many other leukodystrophies. So the sooner we can diagnose, the sooner we can try to intervene. So I just want to end by thanking all of you for the support and willingness to engage with us, because this is the only way that we can get potential treatments to market, and to just say that, for us, patient is what ultimately matters, and that's what we want to make sure that that we work tirelessly, because we know that patients cannot wait. So thank you so much, and I'm happy to answer any questions. Okay, thank you.
Okay, thank you. Our next speaker is Dr. Pamela Folds. She's the Chief Medical Officer of Spur Therapeutics, and will provide us with a program update on SBT 101.
So thank you, everybody. You've probably never heard of Spur Therapeutics, and so one of the first things I'm going to mention is who are we? We are actually a company that has emerged from the SWAN AMN program, has been acquired by a company used to be called Freeline, and all together we are now called Spur. So as we go through all these transitions, you'll hear a couple names, but this is still the SWAN Bio AMN program. So that's very important. The program doesn't change, the investigators don't change, the data we collect doesn't change, just the name you're going to see on some of our materials. So a little bit about SPUR. We do have multiple programs, and what's really nice about this acquisition is it does bring together a couple of programs of gene therapy in the central nervous system. And so it's really nice that we can learn from each other, we can learn about side effects, we can learn about treatment options and how to deliver the product, and so it really brings together some nice learnings. We also are looking into subsets of Parkinson's disease, a rare disease called Gaucher disease, and ultimately hoping to look also in some larger programs such as cardiovascular. So really looking to even larger programs than only the rare diseases, seeing if we can get gene therapy to larger audiences. So AMN, many of you know, I don't need to talk about it. We've heard a lot about it over the time we've been here. Really important is, of course, that it is a disease that has a single mutation in the ABCD1 gene. And this is a gene that's that produces a protein that is required to deliver these very long chain fatty acids to the part of your cell that breaks them down. Your body is naturally always recycling materials. It is always looking to recycle materials, excrete materials, and so it's really important, all of these sorts of recycling and breakdown mechanisms. And what happens in AMN is you're missing an important protein that brings these very long chain fatty acids to the area of the cell that breaks it down. And so they build up within the cells, and then they cause cell damage. And what happens in AMN is that it predominantly is seen in the spinal cord, or so nerves within the spine. And so what happens there is you have multiple clinical aspects of that. You have difficulty with strength because it hits some of the areas of the spinal cord important for strength and motor. It also hits parts of the spinal cord that are important for sense of where your body is in a given time point. So thinking about how when you walk, you need to understand when your foot hits the ground. You need to understand what your other leg is doing while you're walking. There's a lot that your body naturally does to balance itself through multiple ways. And so in AMN, one of the problems that we have or we see is that patients lose some of that ability. And so they can be off balance. They can have difficulty with stairs. They can have a lot of trouble with understanding sometimes where parts of their body are. And so you can imagine balance is a major impact here. And so our program is SBT 101, and it is a program that we are currently enrolling and dosing patients in right now. You've heard a lot about gene therapy, and I'm so glad you showed Dr. Eichler's slide about what are all the different gene therapies. This is in the category of the AAV gene therapy. Where we deliver the gene, the healthy gene, a copy of the healthy gene to the person with AMN so that it can make the protein that your body needs. And so the goal here is that it is delivered by, and I think we even used this slide in some presentations over the last few days, is it is given into your spine. Give it into your spinal fluid over a slow period, and you get that infusion. And what happens is the healthy gene that you need is delivered to multiple cells within your spinal cord so that those cells can start making the protein it needs and clearing out the buildup of very long chain fatty acids and continue to allow new very long chain fatty acids to be broken down. The AAV vectors, we can certainly, there's whole programs on just AAV vectors, so we won't talk specifically about that. But very importantly is making sure that it gets to the specific cells within the spine. So that is highly important. We've seen some really nice data in our animal models, and so looking into the humans now is very exciting. And what's also nice, and you might have heard about this in some other presentations too, is that the AAV therapies don't integrate into the genes your otherwise healthy or the rest of your gene makeup. And so it sits outside and it makes its material outside. And so that does potentially lead to a reduced risk of cancers associated with this. Obviously, there's work we need to do to look at the safety, but that is the benefit, the positive side of AAV gene therapies. And we've seen that in clinical studies, certainly within, also in our non-clinical programs. So our SBT 101 has 2 programs. A lot like Vigil is having natural history data is really important in so many of these areas. Many physicians, certainly many patients and families understand what they are going through. In a clinical trial setting, we have to find a way to demonstrate that these products work in people who have somewhat various levels of disease or how they progress or how they look. And that can get pretty complicated in terms of statistics and showing outcomes and how do you demonstrate a product is working when every person is a little bit different. And so having an understanding of how people progress in their disease, what it looks like for different people, is so vitally important. We also have the second study, which is the interventional study called PROPEL. And what's important too is you'll probably find in almost any natural history study that comes through, certainly within industry, is that you collect a lot of the same data that you would collect in the treatment study, so that you can really have some comparisons here of what is going on and if there is an impact of the therapy. So in our clinical trial, what's very hard is, and we hear this a lot from patient organizations, is why can't you include everybody at the start? And that's always one of the hardest places is in clinical studies. You certainly, you don't know if it's safe yet. Many patients and families come to us and say, I don't care, I want to be in that study, and we understand that. We do have to go slowly. Regulators clearly oftentimes want to see data after, or hear, or want us to look at data after every single patient. We also want to make sure that we're looking at patients where we can see a difference. And so starting with a very, very small patient pool or a small level of range of disease tends to be where many of our trials start. And I know that is very difficult for the community, and certainly in the AMN community right now, we are including just men, and we hear often over and over, what about the women? And we have not forgotten the women, and we will certainly be considering them as the program expands and we go into further studies. So our natural history study is fully enrolled, and we are looking at the data. We're really excited to continue to have that data. We've spoken to a number of different centers just this past weekend about how we can use some of our natural history data with their natural history data. How do we merge this all together? Because the power of natural history is the numbers. It's a numbers game, and so that's where the power comes. So how can you bring this data together to really understand what's going on? And so we're excited about this and to continue to work with other universities and to continue to share the data we see. The phase 1/2 study is called that because it is the first in human. We are giving it to people, of course, with AMN. So it's a very straightforward study, is that everyone knows they get it. We are looking at 2 different doses, and we are simply giving it to every patient and seeing how they do. Just like all gene therapy studies, there is a minimum requirement of follow-up for at least 5 years, and that is required for all gene therapy studies. Because we all need to understand the long-term impact, not only from efficacy, but clearly from safety as well. We have 2 active sites. We are currently dosing, so we have dosed 4 patients so far. Our 5th patient is teed up and ready to go, and we keep our fingers crossed that everything comes together, because as many of you know, these are not simple studies or simple infusions. As I mentioned, we've got 2 doses that we are looking at. You might have heard, and certainly the community, the AMN and the ALD community may have heard the unfortunate news that one of our patients died within the study. And this was very sad, very difficult. This is hard on everybody, unrelated to the product, unrelated to the program. But these things do unfortunately happen, and so just wanted to share with that. And we continue to work, obviously, with the centers to learn what we can, but not related to the product. So what are we doing? So we continue to gather data. Certainly the natural history data, we'll share that. Importantly, as we understand data, we are hoping that we would finish dosing by the beginning of next year, and so that within the first half of next year, we would be able to come out and talk about certainly safety data, any early data we have, so that's a really important component for us. And then all of the work that goes into being ready for what's called a phase 2 or 3, which is the registrational study, which is what the regulators say. These are larger patient pools. You tend to look at a little bit broader of a disease state, different range, different types of people. And this is what you need to bring to get it approved and out to the larger audience. And so, as we've heard, and we'll probably hear from other groups, the key here is speed to getting it to everybody that is appropriate for. And so a lot of work we do is how do we make that happen? How do we get fast to get it to everybody? And that's some of the work that across the board in industry we work on closely. So that is us. Spur. It's okay if you call us Swan for a while. We'll accept that. And thank you very much. Yeah, if there's questions. Nope, great.
All right, thank you. So our next speaker is Dr. Abby Collins, the Medical Director of Clinical Development at Ionis Pharmaceuticals, who will give us an update on Alexander disease and Pelizaeus-Merzbacher disease clinical research programs.
So I'm Abby Collins, and I'm going to be talking to you today about Alexander's disease. I apologize that I can't be there in person. I have COVID, and so decided it would not be appropriate for me to attend in person. I'm going to be talking today about Alexander's disease and our Pelizaeus-Merzbacher or PMD programs. These are my disclosures. And I want to talk about a few disclaimers for the information that I'm going to be presenting today. So this material contains scientific information about investigational medicines that are not approved yet by the US FDA or any other regulatory body. But these investigational medicines are being studied right now in preclinical and clinical trials. So importantly, the information contained in this presentation is not medical advice, and any and all decisions about care should be directed to and discussed with your doctors. So who is Ionis? Ionis is a biotechnology company that was founded in 1989. So we've been around for a while, and our headquarters are in Carlsbad, California. We're a company that primarily looks at RNA-targeted medicines, although in recent years we've been advancing into new approaches into gene editing. We have currently 5 marketed medicines, and we have a leading pipeline in neurology, cardiology, and other high areas of patient need. And some of our medications within the neurology areas include Qalsody, which was approved for ALS, Spinraza for spinal muscular atrophy, and Wainua, which is used for TTR polyneuropathy. I'm going to now talk about our Alexander's disease program, and just a brief overview about Alexander's disease. This is a rare genetic progressive astrocytopathy. It affects the astrocytes in the brain, and those are support cells within the brain. And this often presents as a leukodystrophy in children and as a neurodegenerative disease in adults. It is an extremely disabling and ultimately fatal disease. Current approaches to disease management in patients can mitigate symptoms, but do not address the underlying cause of disease. So we have a clinical trial going on in people with Alexander's disease, and the investigational medicine is called zilganersen or ION373, but the generic name is zilganersen. This is targeting RNA, and it's designed to reduce the production of the GFAP protein, which is the mutated protein in this disease. The purpose of our trial is to evaluate both safety and efficacy in improving or stabilizing function across the full range of affected domains in people with Alexander's disease. So this disease can affect the nervous system, it can affect development, communication, swallowing, can affect the autonomic nervous system, often has GI symptoms as well. may have sleep apnea, and a whole variety of symptoms. And so we have a number of endpoints to look at not only gross motor function, which is common to many patients in this condition, but also secondary outcome measures to look at most bothersome symptom as identified by the individual patient, and then scales to look at people's function and quality of life. In terms of the study design, we have a 60-week double-blind control treatment period, followed by a combined 180-week open label treatment period. So there's a very long open label in this study, and we're looking at multiple doses. We have 13 recruitment sites in 8 countries listed here. And I do want to mention that community member input was critically important to the design of this trial and obviously to the execution of this trial. We couldn't be where we are today without the input of the community, the participation of the community, the sites, and the PIs, many of whom are at this meeting. Before I move on to the next slide, I also just wanted to highlight a unique aspect of this study, which is its phase 1 to 3 design. And what that means is that all usual phases of development, looking at safety and tolerability, which is typically phase 1, dose finding, which is typically phase 2, and efficacy, which is typically phase 3, are all being combined into one simultaneous study in patients with the disease. So we're not doing these sequentially, looking at safety and tolerability first, then looking at dosing, then looking at efficacy. We're doing it all at the same time. And what that means for this study is that we do not have an interim analysis until all patients get to the end of the double-blind treatment period to look at the data generated for safety, tolerability, and efficacy. This is a little timeline about our clinical trial. So we started enrolling in June of 2021 in our first cohort. In June of 2022, we started our second cohort called Cohort B, and that's at a higher dose. And then we opened a third cohort in October of 2023. And this is a sub-study, an open label sub-study wherein everyone gets zilganersen for very young children under the age of 2. And that sub-study will continue to enroll through 2025. Now, if you look on ct.gov about our study, you're going to see 2 different dates in there, the estimated primary study completion date and the estimated study completion date. And these can be a little bit confusing, so I'm going to spend a little bit of time talking about what these are. The estimated primary study completion date is when the last person enrolled in the double-blind treatment period completes their final study procedures at the end of that double-blind treatment period. We're still currently enrolling this trial, and we expect to complete enrollment within the next weeks to months. And so given that we're still enrolling, the last patient that enrolls has to go 60 weeks into the trial until they get to the end. And what that means is right now we're estimating that our primary study completion date will be in the third quarter of 2025. This information has not yet been updated on clinicaltrials.gov, but once we enroll our last patient, we will update that information so that we can have an accurate date for everyone. Now, after the last patient in the double-blind treatment period completes their last visit there, we then can look at the data and analyze the data to see if we have safety, tolerability, and efficacy. And at that point, the results of the data will be released to the medical community when appropriate. So I can't give an exact date of when that's going to happen, but it will be after that last patient finishes their last visit. And we need a couple of months to be able to clean the data, lock the data, analyze the data before we can share it. That's not the end of the study, though. So as I mentioned before, there's this 60-week double-blind treatment period. And then for the main study, everyone has the opportunity to continue on drug for another 180 weeks. And once the last enrolling patient completes the last visit in that open label and then the follow-up period, that's when the study ends. And our estimated study completion date is that point in time. And currently we're estimating that to be the third quarter of 2029. But that's why there is this big difference in these dates. It's because the end of the double blind comes much sooner than the end of the open label. I wanted to thank all the caregivers, patients, and site staff for the ongoing contributions to this study, and we hope to be able to provide an update soon once we've completed enrollment. I'm going to switch and talk about our program in Pelizaeus-Merzbacher disease, or PMD. This is a severe X-linked recessive leukodystrophy, so it primarily manifests in males, and it's also a primarily neurologic disorder affecting the myelin in the brain. PMD is caused by duplications or pathogenic variants, also known as mutations, in the gene that controls the production of a myelin protein called proteolipid protein 1, or PLP1. In patients with PMD, there's too little to no myelin in the brain. And treatments that are currently available may manage symptoms. This is the common theme for leukodystrophies. But as for many other conditions, there is currently no disease-modifying therapy yet available to patients. So for our research program for PMD, we have 2 studies. We have the natural history study, and then we have a phase 1b open label study of ION 356. So I'll talk about the natural history study on the left first. This study started enrolling in 2022 with a goal of enrolling 20 people with PMD, and each person enrolled will be followed for 26 months. Many industry partners who have presented today have already talked about the importance of natural history studies to better understand disease progression, as well as to be able to look at biomarkers and neuroimaging to see how the disease changes over time. Importantly, then, when you introduce an intervention such as ION 356, you can measure whether that investigational therapy has an impact on the disease. And it often takes much longer to see clinical efficacy than it does to see impacts on neuroimaging and or biomarkers. And that's why these natural histories are so crucially important, especially in very rare diseases where that information may not be already available. The recruitment sites are open in Australia, France, Germany, and the Netherlands, and additional sites are anticipated, with the estimated end date of the natural history study being in 2025, but again, this is subject to change based on enrollment. Now, the interventional trial called ORBIT is a first-in-human study of ION 356, and ION 356 is the name of the investigational therapy. This is also an RNA-targeted investigational drug designed to reduce the production of the PLP1 protein in the brain. And like zilganersen, it's given intrathecally into the spinal fluid. The primary purpose of this study is to assess safety and tolerability in children and adolescents aged 2 to 17 years of age with classic PMD due to the PLP1 duplication. We anticipate 24 patients to enroll. And as opposed to the Alexander's disease trial, there is no placebo or sham or control in this study. It's open label and everyone receives the investigational therapy ION 356. All participants need to be able to undergo procedures to collect cerebrospinal fluid through a lumbar puncture, blood tests, and the neuroimaging in order to be able to participate, and additional inclusion and exclusion criteria apply, and those are listed in the ct.gov listing. Our first recruitment site opened in the Netherlands in in the Netherlands and France in April of 2024, and additional sites are opening as we speak. And I believe that a site in the US had their site initiation visit just within the last few weeks. We anticipate completing enrollment in 2025. Again, the study timelines are subject to change based on how quickly enrollment goes. And that's my last slide. I'm going to stop sharing, so hopefully I can hear and take any questions.
As mentioned, I'm Senior Vice President of Clinical Development at ASPA Therapeutics, which is part of the larger umbrella organization known as BridgeBio Gene Therapy, which is in turn part of an umbrella organization called BridgeBio. I'm the medical lead for our AAV gene therapy program for Canavan disease. I'd really like to thank the organizers for the opportunity to speak at this conference and to give such a fantastic meeting. In addition to our partnership with ULF, we also have a number of other very valued advocacy partners around the world who are with us in our fight against Canavan disease. And of course, the most profound thanks need to go out to the families, the children with Canavan disease. They're the ones we strive to work to help every day. So a little bit of background about Canavan disease. It is caused by mutations in a gene called ASPA that provides the instructions for production of an enzyme called aspartoacylase, also known as ASPA, hence the name of our company. The job of ASPA is to break down a very common compound found in the brain called N-acetylaspartate, or NAA. Absent ASPA enzymatic activity, the levels of NAA rise to astronomically high levels. And this is really diagnostic of the disease. And for reasons that still aren't completely understood, this lack of ASPA activity leads to impairment in the production of myelin and the impairment of laying down the white matter. So another white matter disease, another leukodystrophy. So the consequence of that is really twofold. With kind of impaired, not quite up to snuff myelin, the neurons, nerve cells are unable to efficiently transmit their electrical signals, and so their function is impaired. And also the kind of insulating and protective function of myelin is also not fully there. And so cells are prone to damage, to degeneration. So it's kind of a neurodevelopmental disorder in that the developmental myelination is impaired, and also a neurodegenerative disorder that's accompanied by atrophy as well. Now, a little bit about the diagnosis and features of Canavan disease. Just to start with the epidemiology, like so many leukodystrophies, Canavan disease is ultra rare. The epidemiology is not well understood, but an estimated 1 in 100,000 live births. The usual time it comes to be noticed clinically is in the first few months of life, when children are not gaining the developmental milestones that would be affected. And diagnosis is usually made by certain clinical signs, which I'll run through in a moment, as well as this elevated NAA that can be detected in the urine. It can also be detected in the brain by a specialized imaging technique called MR spectroscopy. We'll talk a little bit about that later. And then a genetic testing, identifying disease-causing mutations in the ASPA gene. And 2 mutant ASPA alleles are required for development of the disease. So some of the clinical signs, we talked about this developmental delay. Children's development is stalled at a very, very early age, in the first few months. And some characteristic issues that they have, head control, which is acquired pretty young in typically developing children. They cannot even gain head control. Some cannot even visually track. Sometimes the initial sign that families notice is nystagmus, so abnormal eye movements. Their heads also are unusually large. This is one of the kind of classic features of Canavan disease. So that makes head control even more difficult to attain. Sometimes also in infancy, families will notice their child is unusually irritable or has sleep disturbances, etc. They can have feeding difficulties, breathing problems as well. Many children, but not all, have seizures. They can have low tone in their trunk, so kind of floppiness centrally, and often will develop spasticity and contractures in the extremities over time. So this is affects many parts of the body and many systems and functions, and it really is a 24/7 caregiving situation for affected families. So what are the interventions or measures or supportive care that currently are available? There is no disease-modifying therapy for Canavan disease. We talked about how stalled they are at a very early developmental stage. They require both supportive care around the clock as well as extensive medical care because of their seizures, because of their spasticity. Often feeding tubes and nutritional support. They tend to be young and small and underweight. Ultimately, they will need breathing assist. They'll need cough assist devices to help. Physical therapy, of course, because of contractures and to enhance kind of mobility and mobility aids. So I'm getting into a part of the talk that it's nice to be at this part of the program because the previous speakers have done such a lovely job in introducing many of these concepts. So what I'll do is I'll touch very lightly on some of them. So talking about our natural history study, the case has already been made very eloquently about the importance of natural history data in the context of doing a clinical development program in a rare disease. Our particular natural history study is called CAN-INFORM. There were some natural history data available earlier, but this is really a larger, more systematic study that's intended for two things. Obviously, one, to understand better about the disease, to know what better could look like with a successful intervention. But then also, as was alluded to previously, to provide a control group. This is not a disorder that one would want to need to do a placebo-controlled study design with. And so having very rigorously collected data about function, about developmental parameters in affected children, then serves as a benchmark. If you do the same assessments, we're doing the same raters, et cetera. So tremendous uniformity across the natural history study and the interventional study. So those data really can be comparable and used legitimately for comparison. This is just a little sketch of our, oops, golly. We try to go this quickly in our development program as well. Okay, so we're really, really fortunate in this ultra-rare disorder. We've enrolled 65 participants in 17 different countries with COVID in the middle of all of this. Right now we have 2 sites, one in the United States with Dr. Eichler, who's been very prominent in the field as well as at this conference. And then also with Dr. Annette Bly at University Clinic in Hamburg. We're about to bring Dr. Alex Fay and his team at UCSF at Benioff Children's in Oakland on board as well. So now to our interventional study, CAN-ASPIRE. I have to give this disclaimer. These are preliminary data. We do not know if the early kinds of biochemical changes that we're seeing will translate ultimately into clinical benefit for the patients in our study. There's a lot more to be learned still. This is totally investigational. It has not been approved. It's not been determined to be safe and effective by the FDA or any other regulatory agency. So with those disclaimers up front, let me talk with you a little bit. And I'm going to skip over this. Everyone now, I'm sure, is very familiar with this concept of AAV-mediated gene therapy. In our case, obviously, it's a copy of the ASPA gene. We're giving it via AAV9, which has tropism to the brain as well as to other parts and tissues of the body. We are giving it intravenously. Now, for some sort of CNS-targeted gene therapy candidates, more direct approaches, for example, into the cerebrospinal fluid might be the route of administration, or even kind of sort of more into the brain itself. Here we have some evidence from non-human primate studies that giving intravenously actually gets better into deep brain structures. And so that has obviously tremendous advantages in terms of the kind of burden and invasiveness and tolerability of the administration. So a little bit about eligibility. This is the disease that declares itself very early in life. There's kind of a window of opportunity, if you will, for potentially impactful therapeutic benefit. And that's early, during that time when developmental myelination is still ongoing, a time when neurodegeneration hasn't begun to set in and really with irreversible changes. So for the best kind of benefit risk, especially early in a program, it's important to go young. So our age limit now is for children to be dosed no older than 30 months of age. Of course, they need to have an established diagnosis of Canavan disease, clinical characteristics that are classic for the disease, biochemical evidence in terms of their elevated NAA, imaging manifestations of impaired myelination on MR scanning, and then also genetically 2 mutant copies, pathogenic mutant copies of the ASPA gene. But they need to be strong and healthy enough for to the rigors of the study, if you will, and so otherwise pretty medically healthy. Also negative for anti-AAV9 pre-existing antibodies, because otherwise their immune system would essentially clear the investigational gene therapy before it has a chance to do its job. This is mostly everyone in this very young age group is negative. It is a long haul, as was alluded to before, with this 5-year follow-up period that's demanded by regulators at this point. This is such an evolving field. To really understand those long-term effects requires many years of follow-up. So also, as with any exposure to a new virus, immune response occurs. This is a lot of virus being given all at once, so much more kind of a impact than a naturally occurring infection. And that can sometimes evoke a rather, shall we say, exuberant immune response that can actually cause collateral damage, if you will, within the patient who's receiving the virus. So there's we're using corticosteroids as kind of a broad spectrum anti-immune preventive intervention, starting before they get their infusion of the investigational gene therapy. And we're continuing it over a 3-year period. So this is a pretty aggressive and long-term coverage. But some of the inflammatory events that have been seen in AAV gene therapy, they can kind of flare up relatively early, maybe sometimes even in the first week, but sometimes also later in the 4 to 6 week time frame after infusion. So from experience, we have landed now on this more higher dose and longer duration of corticosteroid coverage in our patient population. So it's a busy study. There are lots of different study procedures. We want to understand so many different levels of what BBP-812 may be doing in dosed participants. So there's a series of motor and kind of developmental tests that are administered by very trained, highly trained research physiotherapists. We are also looking at biomarkers. In this case, ASPA is the enzyme, NAA is the substrate. The substrate goes way high when there's no ASPA activity. So it's a very direct, kind of logical biomarker to look at what we would call a pharmacodynamic effect, or is the actual ASPA enzyme being produced, and you can see that because the activity is there and the NAA levels are going down. We're looking at that in different compartments. I'll talk about those data in a couple of minutes. But imaging as well. I mean, this is a disease of myelin, and it has very characteristic, very profoundly abnormal looking myelin on MRI in affected children. And we've been, well, I won't talk, I guess I will talk about the data a little bit now, but we're following with serial MR imaging at month 3, 6, 12, and then in the long-term follow-up period as well. And we've been beginning to see some really exciting potential evidence of new myelination happening in patients who have received BBP-812. But of course, that requires anesthesia. We're also collecting cerebrospinal fluid to look at NAA there, and also for safety. So there's a sedation involved, et cetera. And there's lots of blood draws. We're very, very vigilant about safety, particularly in the early days after administration of BBP-812. One of the things we're particularly sensitized about is a phenomenon called thrombotic microangiopathy that nobody had ever heard of, I don't know, 5, 6 years ago, but now is something that has come up in a number of different programs, a number of different patient populations, different viral capsids. It happens in adults, it happens in children. It's always when the AAV is given IV. But this is mediated by the early part of the immune response, the so-called innate immune system that just goes in there guns blazing irrespective of what the stimulus is, and the complement system in particular that just goes to town. And again, it's a matter of collateral damage. If that stays rip-roaring level of activity too long, there can be damage that shows in the form of decreased platelets, clot-forming cell fragments, Also, damage to red blood cells. You actually get clots across the blood vessels that shred, literally shred, red cells going through them. And then ultimately, organ damage, which is the thing that one really is concerned about, particularly in the kidney. So there's been increasing interest and awareness about this, the risk of this phenomenon. So much more frequent kinds of lab tests done early in the first 2 weeks after dosing in order to appropriately have surveillance for this phenomenon. It's rare, but it's quite consequential when it happens. So our current study sites, Dr. Eichler, as everyone knows, at Mass General. Also Dr. Alex Fay, Benioff Children's in the Bay Area. And a local physician now just came on board, Dr. Jen Rubin of Lurie Children's right here in the Chicago area. So a little bit about the study, kind of the structure of the study and where we are in the progress of the study. So families who are interested in potential participation for their child can register online, and we have a coordinator who is not part of the day-to-day. She's somewhat else. She will deal with the private information of families that they'll provide. The investigators then, kind of behind closed doors, at least figuratively in this age of Zoom, will review the prospective candidates for those that they think are most appropriate to invite in for screening. This is something we as sponsor have nothing to do with. They're the experts. They're the Canavan disease experts. So patients will come in for a screening period of about 6 weeks, and then inpatient for a few days for the infusion of BBP-812, watched for a few days, but then tend to stay near the area, and this is something we as sponsors support for them, because there are so many visits, and we do have patients from multiple different countries around the world. But the kind of immediate follow-up period is a year, again with imaging, with motor testing, biomarker testing, et cetera. And then this longer-term follow-up period for another 4 years. It is, as was talked about in one of the previous talks, this is a dose finding as well as what we hope will then become our pivotal, our registrational trial. So this is a seamless design. Right now, we're in this dose finding part. We have explored the initial dose in 8 participants, and now we've dosed 2 at the higher dose. As also was alluded to, there's this required kind of cadence with a interval of observation between dosing each successive patient, again for safety. Once that has kind of been gone through and the safety looks acceptable enough, then that cadence can pick up. But it is incredibly frustrating for families to kind of have to wait for this, what feels like an arbitrary period of time. But it is all because safety is job one for any clinical trial. So we're hoping to get to that point next year, as well as have this morph into what will be our registrational study, that we're going to put the data from that part in front of FDA, for our candidacy for approval. And we are doing an accelerated approval approach. Again, the most efficient. We have a very plausible biomarker in terms of the NAA levels going down, that the enzyme is being expressed and it's doing the right thing. And if we have supportive clinical data, which we're starting to get, I'll show you in a minute, hopefully that will really make a compelling case for accelerated approval with FDA. So, speaking of biomarker, we have followed NAA levels in participants in urine, obviously the easiest place that you can measure NAA levels, as well as in CSF, less frequent sampling as you can imagine, and then also in brain MRS. What I'm showing here is right frontal white matter because it's a white matter disorder. But the pattern is the same throughout. You can see the very, very high levels of NAA in participants coming into the study. You see the little kind of pinkish red dotted line at the very bottom, right above the x-axis. That is the range for unaffected individuals. So you can get a sense of how hugely elevated NAA is in these patients. So they all come in with these astronomically high levels, but then after receiving BBP-812, there is a pretty profound and rapid decrease. And what we've been noticing as we follow over many months and now for some patients years, there's durability in that effect. They're not getting down to the normal level, but they hit kind of a plateau, but then they're able to maintain it over many, many months or years. The other thing that we've noticed is kind of of particular interest is the levels to which they fall are within this band, within this range that's characteristic of milder phenotype Canavan disease. There are a very small percentage of patients who have Canavan disease, who have mutations, they have developmental and motor impairments, but the phenotype is much less severe. And it may be because their mutations allow a little bit of ASPA residual activity, but in any case, they have much lower NAA levels. And so we're getting patients who have severe Canavan disease to have their NAA levels now in this milder range. That does not necessarily say that clinically they're going to end up having a mild phenotype. We just have to watch longer to understand what the clinical correlates are of this leading indicator of NAA decrease. So now a little bit about our clinical data. This is just a snapshot. We have so many different scales that we're using that measure so many different aspects of function in these children. But this is just a little bit of an overview. We've identified certain skills that most Canavan disease patients haven't been able to achieve, and yet are highly clinically meaningful, very meaningful for their function, and maybe even stepping stones for further motor and developmental gains subsequently. So these consist of visual tracking, head control, rolling, weight bearing on the legs, sitting, and also use of the hands or control of the hand use. And the dark green cells indicate progress since dosing. And this is, an increase in an item score. We don't specify which particular scale this is being noted on, but they have shown an improvement since prior to receiving BBP-812 on an item that captures that particular function. It doesn't mean they've gained full function, but they're showing some improvement. The other thing we're capturing is maintenance of existing skills, and that's the lighter green. So typically, even if a Canavan patient gains a skill, they typically then regress and lose it. So the ability to maintain over many months after BBP-812 administration, that's also very noteworthy when individuals might otherwise be losing the skills that they had had. The gray boxes represent that they didn't have the skill before and they don't have it now. And we, in principle, had a darker gray box when an existing skill was lost. But fortunately, so far, that is not something we've seen. No patient has lost a skill they came in with and then subsequent then lost it after BBP-812 administration. So this is all something that makes us very excited in the promise of our investigational gene therapy for Canavan disease. Also talking a bit about safety. So as you can imagine, every participant has had different kinds of adverse events. Most have been mild or moderate in severity, and most have not been attributable to BBP-812. There have been some serious adverse events, again, most of them not considered related to BBP-812, except for a couple, one that was possibly and one that was related relatively recently, one that emerged. So to date, the overall profile of BBP-812 safety in our Canavan patient population is very characteristic of what has been seen with other systemic AAV9 programs. So that's been reassuring to us so far as well. So of course, I have to end with a tremendous thank you to the Canavan patients and their families. And there is our scientific founder, Dr. Wang-Ping Gao, whose pioneering work really made this all possible, and to whom ASPA owes a tremendous debt of gratitude. So thank you very much, and happy to answer questions. No questions? Great, thank you. Oh, I'm sorry, I'm in the blinded.
I'm sorry. So the dark gray box that was skill regression after you started the study, there was none of that. Would there untreated children with Canavans? They would have had skill regression without the treatment for sure, right?
So
I mean,
we've
that was
looked-
typical.
That's a great question, and I didn't talk about the natural history for context, but we've been looking very deeply into our natural history data, again, to kind of make that case, you know, what do they look like, and what does this mean in comparison to what would typically happen? And really, the take-home message about all of that is most children function at a very low level. Most don't gain head control. Many don't even develop visual tracking. Sitting independently is almost unheard of in classic severe Canavan children. We've omitted some who have genotypes that have been published to be known to be associated with this milder form, this very rare mild form. They're not part of what we consider our natural history comparison group, because our enrolled patients in the interventional trial are also intended to be, you know, severely affected classic patients. So really, it's remarkable how flat, how at floor, so to speak, the children in the natural history study, kind of in the same age range, are behaving. So this really is a conspicuous difference from what natural history has been showing us. Thank you.
Thank you. Our next speaker is Dr. Arun Mistry, who's going to, the Chief Medical Officer of Minirix Therapeutics, who will provide an update on the CALIX study.
Thank you very much, Dr. Renaud. And thank you for the ULF for inviting me to give an update for you on leriglitazone and, very importantly, the CALIX study. Right. So first of all, introducing leriglitazone and introducing Minirix. Minirix is a small Spanish company based out of Barcelona. I'm not Spanish, I'm English, and I'm the only person sitting in the UK in the rain most of the time. We have one product called leriglitazone, and this is not a gene therapy. This is an oral suspension which comes in a bottle and is to be taken once a day. It's a drug class called PPAR gamma agonist, and this is quite an old type of drug, typically used in the past for diabetes. However, what we have seen has some very important disease-modifying actions in terms of reducing neuroinflammation, reducing demyelination, and also improving the mitochondrial activity, the energy-giving part of the cell. We have activities with the EMA in Europe and the FDA in Europe, in the US, and we also think with the multimode of action, which is not specific to any one disease, we can look at other indications and diseases with the product as well. So just want to try and simplify something right at the beginning. We're looking specifically at the moment in ALD at cerebral ALD. And if we first of all just think about this diagram, which is a nice simplified diagram of 3 boxes and some MRI scans, is patients with adrenal leukodystrophy are born with no lesions that are visible on MRI scans. But at some point, typically in childhood for the boys, and a third of boys can be affected in childhood, they have non-enhancing lesions, which means that the blood-brain barrier is intact. And these can further develop into gadolinium-positive enhancing lesions and with rapid growth as well. So this is cerebral ALD. And this is what we're trying to manage and treat with leriglitazone. And you can see in the MRI scans, this is a non-enhancing lesion. This is a clean MRI scan. And then as the disease progresses, this will become an enhancing lesion with increased growth as well. And that's what we're trying to treat. MRI scans precede any clinical changes. So the scans are very important that they are done regularly for screening for cerebral ALD, both in childhood and also in adulthood, which is very important for the Calyx study that I'll take you through. As these lesions grow, there's more damage in the brain, and then this eventually leads to clinical problems, and this is the NFS, which you may have heard, which is the Neurological Functional Score, and MFDs, Major Functional Disabilities, which appear much later than the MRI scans show any problems. So the MRI scans are very important in finding patients who have got the disease starting in their brain. When the disease does progress, I think you're all aware, this without treatment with bone marrow transplant, typically, if identified early enough, will lead to death. Bone marrow transplant really is the gold standard, both in pediatrics and adults. But we know it's invasive. It can cause both more morbidity in terms of complications, and also can be life-threatening as well, with complications in graft-versus-host disease, etc. So it is quite a serious procedure and has to be managed very carefully. Often patients are diagnosed too late, especially in the rest of the world. I think it's fortunate now in the US you have newborn screening. It's now started also in the Netherlands, and there's some little pockets in Europe, but I think it's pretty unique here in the US that you have newborn screening, which identifies patients early, and they can then be monitored. But there's still a cohort of patients, boys and men, who are not receiving regular MRI scans. So patients can be diagnosed late. There needs to be time to find a donor as well for bone marrow transplant. And really important in men with AMN already, that's ongoing, and the coincidental occurrence of cerebral disease, there's less experience. The men are more fragile because of their ongoing AMN disease. Some of you may have heard that from Dr. Lund yesterday as well. So it becomes much more problematic in men to do bone marrow transplant. In the US, of course, you have gene therapy for the boys, where bone marrow transplant is not possible. So I think that's really good news for the community here in the US. So what am I going to take you through is some of the clinical data and work that we've done so far. Some of it's ongoing, some of it's completed. And we'd like to first of all say a big thank you to the whole community in helping enroll patients into these global studies, which isn't a small feat. I think the advanced study was the largest study in ALD that's been performed, with 116 patients enrolled. It was a study for AMN, looking at the 6-minute walk test, which unfortunately we didn't succeed on. However, there were some very important findings in cerebral ALD, which I'll take you through shortly. And this is a published paper from Professor Kohler in The Lancet. Also, I'll take you through a couple of slides on the Nexus study. This is a pediatric study, which is ongoing. It's an open label study with 23 patients. And I'll be presenting some data from the interim analysis, which was at week 24, and the final analysis is not too far away. And then finally, just a brief update on some recent compassionate use data, which has been published in a journal called Brain, which is really important. Okay, just going back to those 3 boxes, which really simplify the disease, a very complex disease, but is helpful just to think about it in this way. The advanced study had men with AMN, because that was the primary objective, but also what we found was men with AMN either had no lesions on their MRI scan, or actually came into the study already with lesions which were not enhancing. So the blood-brain barrier was closed. They weren't assessed as being progressive. And what we saw for the whole population in the advanced study is that only patients in placebo over a 2-year period progressed to this progressive CALD form, having the gadolinium enhancement or the rapid growth. No patients in the leriglitazone arm actually had that progression. This was a really important sort of signal that we saw. These results also, after 2 years, all the patients were put onto open label treatment for another 3 years, and the same type of information started coming through over the next 3-year period as well, that leriglitazone was reducing that conversion to the rapidly progressive CALD. And this was further sort of reinforced in our minds and the data, really showing that the incidence of cerebral lesion progression, this is not the progressive CALD, this is just either new lesions, growing lesions, or changes in the gadolinium status of the patients. We saw only 3 out of 77 patients having some level of progression, compared to 8 out of 39 patients in the placebo arm. And then the less score, again, with leriglitazone, there was a reduced increase in the less score compared to placebo, where there was a significant increase. And this was also reflected in another biomarker, which you may have heard about over the last few days, called neurofilament light chain, which is a biomarker of destruction of the myelin sheath. So again, we saw with placebo that the neurofilament light chain biomarker was increasing over the 2-year period and was sort of remaining stable, which is reassuring in a sense, but we'll have more data on that soon as well. And then going to the Nexus study, this is the pediatric study. Here we were looking at boys who were already, in a sense, diagnosed with an MRI scan with having lesions in their brain, either non-enhancing lesions, of which we had 9 patients, or enhancing lesions, where the rapid growth would have been expected, and then we had 14 patients there. Total of 23 patients, and the study is closed now, so we're just really following these patients up. There is good natural history for pediatric CALD, and what the natural history really sort of shows is typically there's a self-arrest rate. So if a boy is diagnosed with having a non-enhancing lesion or even an enhancing lesion, there's a 10% self-arrest rate, so the disease just stops. Nobody knows why, and this is at 2 years. There will be a 10% self-arrest rate. Also, typically, if there is no treatment, there would be an increase in the NFS or the MFD scores, and also increased lesion growth as well. So the less score would increase as well, typically with natural history. What we saw at week 24 with 11 evaluable patients was all of them, in a sense, were meeting continuation criteria. And this was either arrested disease or clinically stable without significant growth. Very early data, but in a sense said we could continue with the study. We weren't saying that this was showing an absolute effect. What we have been doing in the meantime with this same data set is looking at the volumes of the lesions, which is one of the key signifiers of clinically stable or without significant growth. And again, we're actually seeing the lesion growth slow down with leriglitazone compared to natural history, for which there is volumetric data. And here, just to share with you some of the neurological functional scores. This is natural history on the left from Professor Raymond's paper. And here you can see once a patient is diagnosed, there's an increasing of the score quite rapidly if there's no treatment. Here, this is the data from also the same paper showing once you treat with bone marrow transplant, HSCT, the worsening of neurological status really is slowed down dramatically with treatment. Very early data with liraglutide. We have some patients here with up to 2 years of data. At that time point, this is 18 months old, this data, so there is new data coming soon. We were still reassured that there was no sudden rapid increase in both populations, either gadolinium negative or gadolinium positive. So that was reassuring. And then when we look at the LESS score, the MRI imaging, again, what we see here is when patients are treated with HSCT, here on the right, there is some increase of the LESS score after treatment, but then a plateauing. Very similarly with Elicel and Skysona, once there's treatment, there's some continued increase and then flattening. We sort of see the same very early on with all of the patients at the time point of the interim analysis. And then finally, in terms of our data, just wanted to touch upon compassionate use. And compassionate use is ongoing in Europe, where we're able to do this. It's a very heterogeneous population, but what has happened in France is there is a protocol that's driven by the French regulatory body, where adult patients have been going through a protocol with regular assessments. And what has been published just now is that 13 patients have been assessed over a 2-year period by Professor Michel in Paris. And from these 13 patients, 10 patients have remained clinically and radiologically stable. 4 actually have had some level of clinical improvement, which Professor Michel was very intrigued by. 2 patients did decline. 1 patient died of COVID-19 during the data collection. And there were some patients who had gadolinium positive lesions that changed to gadolinium negative. And some of these patients came into this study on the bone marrow transplant list, awaiting the right donor. What we saw, what Professor Michel saw, was 5 out of 5 of those patients actually came off that list and have remained stable off that list, continuing taking liraglutide. So, again, reassuring data and important to sort of give us some confidence to keep going with the clinical trial program. In terms of safety, this is a product which is from a family of drugs which have been used for the past 20, 25 years in clinical practice for diabetes typically. And the things that we would expect do happen, so edema and weight gain. The edema is typically mild or moderate and normally peripheral, so ankle, leg edema, but sometimes some facial puffiness as well. But it's not associated with any concern with fluid retention that's causing any problems with the heart. We've been doing regular echocardiograms whenever there's edema, and we've not seen any issues there at all. And it's reversible once the drug is stopped. In pediatrics, the product is actually tolerated even better with very little in the way of edema, no cardiovascular events again. Patients continue to grow, which is important, and we've not had any withdrawals due to any adverse events in the pediatric population. So what do I want to really talk about, and that's just a quick update, is the CALIX study. This is a phase 3 study in adults with CALD, with gadolinium enhancement. And this is a phase 3 study, randomized and placebo-controlled. So we've been hearing about natural history. And unfortunately, despite CALD, adrenoleukodystrophy, being well understood and known, there's no published data on the adults with CALD. There is some data being developed and ready for publication from Professor Kohler and with help from data from Professor Eichler as well. So that's coming, but that wasn't published, and therefore the FDA have asked for this study to be placebo-controlled. What we're looking for are adults, in a sense, over 18-year-olds, with a diagnosis of cerebral ALD who are not recommended for bone marrow transplant. We need gadolinium-enhancing lesions with a less score of less than 12. So that's wrong, so apologies for that. It should be less than 12. We're looking for 40 patients. 20 would receive the active drug, leriglitazone, and 20 will go on to the placebo arm. We're looking for patients in the US and South America, and we are potentially going to start looking for centers in Europe and also possibly towards Asia as well. It's an adaptive duration, so it's a study that's up to 36 months, but we're able to look at the data at 18 months, after the last patient reaches 18 months. And the primary endpoint is time to death, or for the patient to be bedridden or with permanent ventilatory support. And this is because there is no natural history data that we've had to do, in a sense, what is like very similar to an oncology study, a cancer study. But we think this is something that we will see some positive data on. Of course, we're looking at MRI scans and assessing the LESS score. So, as I've said, it's a adaptive duration study. So it's a placebo-controlled study, so half the patients will go onto placebo, half onto leriglitazone. And then after 18 months of dosing, we can have a look at the data. We don't look at it ourselves. The data monitoring committee will look at it independently. If there is efficacy in terms of a success for the primary endpoint of survival, all the patients would be transitioned to the active drug. If there's no signal for efficacy, the study would carry on as placebo-controlled and for another 9 months, where there's another opportunity of looking at the data. Again, if successful, everybody moves to open label, leriglitazone. If unsuccessful, we continue the study to the final analysis at 36 months. We're able to do these analyses without any penalties. Typically, if you look at data through the course of a study, especially a placebo-controlled study, you have to bring more patients in. The FDA have actually been quite kind to us and said we don't need to look, because we actually have quite an objective endpoint, primary endpoint with survival. The study is running in North America and Latin America at the moment, so we have the typical centers I think you're all aware of, in terms of Baltimore at KKI, Boston at MGH, Minnesota, Utah, Stanford. We also have a center in Florida, and then 2 centers in Latin America, one in Buenos Aires and one in Brazil, the other way around. What we're trying to do, which is really important for the men with ALD and AMN, is really bring them back into routine screening for MRIs, which we know is a pain, especially when the patients aren't being seen by the typical ALD expert centers. So what we're trying to do is offer free MRI scans and possibly genetic testing if the MRIs are positive in any way, which can be performed locally to the patient or at any of these centers. And if the patients are eligible for the study, we will provide free of charge again the genetic assessment as well. There is a website where patients can be referred into called cald-study.com. And what we really want to try and have the support from the community for is really getting men with a diagnosis of AMN to attend for MRI scans. We know this disease in adults is sporadic. There's no prediction as to when it's going to happen. The only way of finding it early is to have an MRI scan. If it's found early, the opportunity again here is for the patient to be assessed for bone marrow transplant. If it's early, that is absolutely an opportunity. If it's too late, that's where the problems of not being able to do a bone marrow transplant happen. So we're hoping that what we're trying to do here getting the community to have MRI scans will allow more patients to be treated with HSCT, with bone marrow transplant. If they're not eligible for bone marrow transplant, another opportunity is the study in terms of trying to help the community, but also a chance where a 50-50 chance of receiving the drug. So I'll stop there and say thank you very much, because this is a big endeavor for the company, but absolutely for the community. Everybody's really engaged with trying to get more men back into routine screening. Thank you very much, and take any questions.
Our final speaker this afternoon is Erin Murphy, the Executive Director of Corporate Affairs for Mirum Pharmaceuticals, who will introduce us to Mirum Pharmaceuticals and their dedicated focus on rare disorders.
Well, I'm bringing up the caboose on the industry updates. Give yourself a pat on the back for hanging in there. Well done. I am very, very pleased to be here to share a little bit about Mirum. Sorry. So we're newer to you all. This is our first time attending as Mirum. Last year, we acquired 2 medications from Travere Therapeutics. You may remember them, had met their team, wonderful team in the past. And we took those over to continue the development and to bring those to patients and to continue to make strides forward and continue the wonderful work that they did. So as we're new here, and I figured, you've likely heard over the last few days about CTX and the wonderful progress with Chenodal and our RESTORE study. I thought I would take a little bit of a break from that and take this opportunity to introduce you to our company and who we are and all that we're dedicated to. So a little bit about our team. We are based in Foster City, California, right in the heart of the Bay Area, in an area that my mom was very concerned about me working because she said if there was an earthquake, it would definitely fall into the water. Alas, it hasn't done that yet. It's a really, really great pocket, very close to the birthplace of biotechnology in South San Francisco. We've got our headquarters there for the US, some employees working there, but we also have a remote team. And then we have 2 offices that operate in Europe, and that houses our development team in Basel, Switzerland, and then our commercial team in Zug, Switzerland. And then we have folks living throughout Europe to make sure we're nice and connected to all that's going on globally and ensuring that we can get our medications to patients. Our team is comprised of about 300. We're growing like a weed. We actually, a beautiful weed, I should say. We actually hit our 5-year anniversary just last year at the end of the year and are continuing to grow our team rapidly. We did a lot of our hiring during COVID, brought wonderful people on. We're able to really hit some fantastic milestones and make a lot of strides forward in our programs, and we're really, really proud of that and excited to continue that work. Our team really has come together with a lot of industry veterans, a lot of people that have focused on liver, on rare disease, and that's something that's so important. We definitely look for a specific phenotype to come work with us, people that are really passionate. Even if you're not working in patient advocacy or in clinical, we have people that are working in finance and human resources that want to do something big and want to be part of something important and making really important strides forward for patients. So you really see that as you come into our halls, and you really get that vibe from the people that we work with. Research is the center of everything, and I think it's important to always pause and really think about why we do that. And one of the most important parts, I'm talking to people that understand this, of course, but really understanding our understanding the disease. So as we look at, especially in rare disease, it isn't one size fits all. Everyone knows that. You may be diagnosed, with the same disease as the person next to you, but it's going to look completely different. And so talking to one person or working in industry for a long time is never going to give you the full picture. So speaking with the patient communities, staying connected to physicians and academia, and talking to as many people as we can to really get a broad sense of what we're dealing with is going to help us find the best treatments forward, create the best programs, and ultimately the best outcomes for patients. We want to evaluate these really great scientific advancements that come through. We want to look and uncover every stone to look at what can be most impactful, because what we're often dealing with are diseases for which there are few or no therapies. I know I've been to many conferences where I've heard families say, please find something, go shop around, talk to other scientists. They're asking whether it's the FDA or companies to go out and find answers. And that is what we all want to do. And I think collectively as an industry, and we look at sort of the trifecta with the patient communities, academia, scientists, healthcare providers, and then industry, we can all do that together and make really, really important advancements happen. And then at the end of the day, we just want to make people better. We want to create that hope. We want to come and inspire and give updates at conferences like this and be able to say, we've made great movements forward. We have exciting data to share with you. And that's always, at the end of the day, the healthier tomorrows is what we want. A little bit about our areas of focus. So I talked about Mirum being a rare disease focused company. That is our heart and soul. We started in rare liver, and we have Livmarli that is on the pediatric side. It's an IBAT inhibitor. It's improved for 2 rare liver diseases. One is called Alagille syndrome. The other is called PFIC. Those are approved in the US and Europe for Alagille, and then the US and hopefully soon in Europe for PFIC. We also have Chenodal that you are likely familiar with, and that is being evaluated for CTX. Traditionally, it was approved for medical necessity by the FDA because there were no other treatments. But I am thrilled to say, so serendipitously, that just on Thursday night, our team filed an NDA with the FDA for approval, and so we are hoping that that will be just around the corner, hopefully in the early part of 2025, that we'll get an approval. That would be our hope. So I always love it when the slide looks out of date and we have a real-time update, so I would give that a massive woohoo. And then we have Cholbam, which is approved for bile acid synthesis disorders, for PBD and CSD, so other rare diseases affecting a number of patients. And then on the adult side, we have an IBAT inhibitor called Volixibat that is being evaluated in PSC and PBC. And we had interim analysis of our data that we shared just a couple of weeks ago and had really exciting positive results. So we're very excited to be able to carry forward those studies into the next phase, which will hopefully be confirmatory studies that will hopefully lead to an approval. Now, as we think about what we want to do and how we want to get there, the voice of the patient truly drives everything that we do. We really make sure that, collectively, we've come together, we have lots of years of industry experience as a team, but as I said before, you only know what you know, and until you have that perspective from the family, from the advocates, from the people that are living with the disease each day, you really can't create a comprehensive program. You can't really do the best work. And so we make sure that that voice is integrated, that we get a lot of feedback, that we're asking the questions, we're listening and learning and repeating, and really infusing that into all aspects of what we do at the company. And collaboration is absolutely key. This is a small smattering of the groups that we work with, which we just find integral to what we do every single day. And this is not just a call twice a year, check in, say hello, see how things are going. We want to keep that conversation flowing all the time because it is really important to understand and also know that the needs of the community are changing. What are you hearing from patients? What are you hearing from families? What are the needs out there? As you think about research, what would be most important? as we think about symptom burden, what is needed. And that's on the clinical side. We also think about as a commercial drug, as a drug comes to approval and becomes commercially available, what do the patient services programs look like? How do we set them up so they really fulfill the needs of the family? Is it that going to the pharmacy is just far too burdensome? Then let's look at an option for being able to mail out the prescription medication. Having staff on hand to answer questions 24/7. Whatever it may be, we want to make sure that we're getting that input and bringing it back and folding it into our thinking at all times. And just to kind of follow in with a few examples, I like to talk about examples because I think just showing that connectivity in the ways that we really infuse input from the community and from the advocates is really important. So something as important as going to the FDA and talking about symptom burden. They hear from industry, they understand, they look at the research, and that's great, but having someone there to say, this is what it actually looks like firsthand from a family, on behalf of the community, that helps to really paint a picture that is connected to what we're doing and to help really understand and characterize better what we're dealing with. We participate, you've heard a lot about the natural history studies. That's often something that we'll do in collaboration with advocacy groups and research organizations. One thing that I love to point out, for us in Alagille syndrome, this is a disease that affects children. It manifests in a lot of different ways. There are a lot of common symptoms that are burdensome, but this impacts the whole family. And there was no way that we could bring what we knew to the table every day and make decisions because the situation is so different for each family. And so what we did was we actually brought in a mom to come work on our caregiver marketing team. She has a really big voice, she has a lot of ideas, she's very well connected in the community, and we offered her a job to come work for us and to be that voice of the patient every day. So we're not just making decisions in a vacuum, but to actually look at her and say, is this correct? Is what we're doing on track? And she pushes back often, and we feel like we've come to so many great resolutions and great ideas because of that. It's just one layer deeper in really understanding and making sure that we're connected. With that, I just wanted to thank you. Again, I'm really excited to be here. I feel very honored to be here. I know I've been texting with members of our team, and they have massive FOMO. They would love to be here to meet everyone, and so I think in the future you'll see many more Miramars here, but we're really delighted and honored to be part of the community, and I thank you so much to everyone who organized this wonderful event. I know I've learned so much. I have a lot to research on the plane home, but thank you for all that you do.
So I want to thank all of our industry presenters today for very clear and instructive presentations. And I'd also like to thank our industry partners for their dedication to the development of treatments for rare disorders, and in particular leukodystrophies, and for their support of this conference. We reached the point where we have one parallel session left that's starting at 4:15. And in various rooms. And so I think this is a good opportunity for me to say thank you to the planning committee for the family conference, and to Diane and Tara at Keeley, Bob and Ron and all of the ULF team for really organizing an outstanding conference. It's really been slick. I thought it's worked so well, and it's been very informative. I would also like to take a moment to thank the volunteers, particularly those who helped keep the children entertained. Bowling yesterday was very fun to watch. Thank you to the volunteers who have been helping. Without you, I don't think we could have made all of this work. The other thing is I'd like to thank the families for coming. We're in this together. And I think listening to all of the industry speakers today and the scientists who have been presenting at this meeting, we're at a point now where we have hope for the future. And so please Enjoy the rest of your day and travel home safely.